eukaryotes, this group of complex cells, are just enormously more complex than bacteria or archaea. Something happened, and we don't really know what it is. So there's a paradox there, and it's a paradox about natural selection itself. So essentially, the problem is that all the complex life that we know, and that includes amoeba and paramecium and so on, is composed of this one cell type. And all those cells are essentially the same. It's very difficult to tell the difference between them. They all share a long list of hundreds of or thousands of traits in common. It only arose once. We all share a common ancestor, and that common ancestor by definition arose once. You could say perhaps there were lots of other origins as well, but there's no trace of it anywhere in biology. So, it's make-believe. It might have happened, but there's no evidence for it. We all share these traits. Prokaryotes, that means the bacteria and the archaea, they show essentially no sign of evolving any of those traits. They have, I showed you the nuclear-like compartment, that's about as good as it gets. So there are steps in the direction, but really nothing very impressive in comparison to what eukaryotes do. Now, if all of those traits, if the nucleus or if sex, for example, arose step by step, as natural selection says it must, and each of those steps offered some small advantage, which I'm not disputing at all, that's what happened, then why did none of them arise in bacteria or archaea? The host cell was an archaeon from the archaea, and there was an endosymbiont, so a cell that got inside it, and that cell went on to become the mitochondria. So we all have mitochondria, the powerhouses of eukaryotic cells. So it must have been something a little bit like this. So this is the one example that we know of today of a bacterial cell. This is actually a cyanobacterium, and you can see there's some membranes in there. These are thylakoid membranes in cyanobacteria, with cells living inside them. We have no idea how they got in there. They weren't engulfed by phagocytosis, because this is not a phagocyte. It has a cell wall. But they're there all right. We just don't know how they got there. But this is the starting point then for the evolution of complex life. I want to suggest that the problem with this is that they are constrained by pumping across their membrane. This is how life started. Bacteria, archaea, all do that in that way. And then they're kind of stuck with it. So what was the good thing about having this situation at the origin of eukaryotes, where you have an endosymbiont? This goes on to become the mitochondria. Well, the thing is that these are cells. It's not just a cell, it's a population of cells, and they can compete among themselves. And when they do so, very often, if you can make more copies of yourself in a given period of time, you will be better represented down in history. And so if you get rid of a few genes that you don't need, for example, you make more copies of yourself in a shorter period of time. And so the competition between these cells in this population living within a cell, they tend to dump any genes that they don't need and become simpler and rely on the host cell for anything that they can do. And in the end, they lost almost all of their genes. We only have 38 of them left in our own mitochondria. Well, there are massive, massive energetic consequences of losing those genes. So just as a quick and slightly silly thought experiment, imagine that you've got a cell and it's got 100 endosymbionts. So it's got 100 bacteria living inside it. And let's say that it has a standard bacterial genome. So it's got something like 4,000 genes, 4 megabases, 4,000 genes. It loses 5% of them. So these are the genes it normally would use for making a cell wall to protect itself. It doesn't need that anymore, and they replicate a bit faster if they lose those genes. So it's lost 5% of its genes. So what's the energy savings that you get from losing those genes from 100 endosymbionts? Well, each of those genes encodes a protein. So few conclusions. Complex cells, the eukaryotic cell, arose just once in 4 billion years. That seems to be a fact. I can say more about that if people are interested. It looks as if energy constraints on bacteria demand some kind of a rare endosymbiosis between prokaryotes. Conflict then between the endosymbionts, between the cells living inside a host cell, that's pretty tough, and it forces cells down populations of cells down particular avenues. It forces you to have sex, for example, or to have two sexes. And so we can begin to predict that the resolution to those conflicts may account for why all eukaryotes share so many similar traits. Now, I want to leave you with this. This was discovered, in case you think it's pessimistic that complex life only arose once in 4 billion years. Maybe it's happening again off the coast of Japan. So this was discovered about 5 years ago, and it's an intriguing title on the paper, a unique organism from the deep sea, prokaryote or eukaryote. They don't know, I don't know, nobody really knows, but let's have a quick look at it. So it's about 10 microns long, which is substantially larger, these are bacteria here, so it's substantially larger than the average bacterium. It has what looks like a nucleus, so this looks like a standard eukaryotic nucleus. It's got some endosymbionts. Perhaps they're not mitochondria, but they could be something like hydrogenosomes, which are derived from mitochondria. It's got a plasma membrane and a cell wall and so on. So to a first glance, this looks like a standard eukaryote. But when you look more closely, well, this is not a double membrane. All eukaryotes have a double membrane around the nucleus. This has a single membrane around the nucleus. All eukaryotes have chromosomes with a diameter of around about 30 nanometers. This has bacterial filaments with a diameter of 2 nanometers. We don't know what those are. There's no genome sequence for this. We really don't know very much. This mottling here, those are ribosomes. No eukaryotic cell that we know of has ribosomes in the nucleus. The whole point of the nucleus is to exclude protein synthesis from going on there, but here it's going on in the nucleus, as well as outside the nucleus. These membranes inside the cell, they are nothing like the membranes that we see, the endoplasmic reticulum, things like that. So this is not a eukaryote, and it's not a prokaryote. I would say that what this is a bacterium that acquired endosymbionts and which is recapitulating eukaryotic evolution. It's become larger, it's accumulating genes, it's got a nucleus, it's going through the whole thing all over again. But we only ever found one of them, and they've been looking for 15 years to try and find another one, and they've not found it yet. So, and they took a section of that, which means we don't have a genome sequence either. So this is how science goes. We're just going to have to wait, and you are as well.